Your gut contains approximately 39 trillion bacteria, more microbial cells than human cells in your entire body. This ecosystem regulates your immune system, produces neurotransmitters, synthesizes vitamins, controls inflammation, and influences your risk of diabetes, obesity, depression, and autoimmune disease. Yet most people's gut microbiome is profoundly impoverished. Decades of antibiotic use, ultra-processed food, and low fiber intake have decimated the diversity that protects health. Repairing gut health is not about a probiotic capsule. It requires a systematic, evidence-based approach to rebuilding microbial diversity and restoring intestinal barrier integrity. Sonnenberg and Sonnenberg's 2014 Nature analysis proposed that Western industrialized populations have created a microbiome extinction event, with dietary fiber restrictions so severe that species of bacteria that co-evolved with humans for millions of years are being permanently lost within generations. This is not reversible with probiotic supplements. It requires sustained dietary diversity to rebuild what has been lost. This video draws from 5 peer-reviewed studies across Cell, Nature, and Physiological reviews, covering the microbiome scope, the role of dietary fiber, probiotic effectiveness, obesity and gut dysbiosis, and the gut-brain axis. The supplement industry has positioned gut repair as primarily a probiotic problem, one solved with a daily probiotic capsule. Zmora et al.'s 2019 Cell study challenged this directly. In their randomized trial, standard probiotic supplements colonize the gut in fewer than 50% of participants, and in some cases actually delay the restoration of native microbiome diversity after antibiotic treatment compared to allowing natural recovery. The microbiome needs feeding, not flooding with single strains. Turnbaugh et al.'s landmark 2006 Nature study demonstrated that gut microbiome composition directly influences energy extraction from food and obesity risk. Germ-free mice colonized with microbiota from obese donors became 47% fatter than those colonized with lean donor microbiota, eating the exact same diet. This established gut dysbiosis as a causal factor in metabolic disease, not merely a correlate, with profound implications for weight management beyond calories. Signs of gut dysbiosis and intestinal barrier dysfunction are often dismissed as digestive complaints rather than recognized as the systemic health issue they represent. Cryan et al.'s Physiological Reviews Comprehensive Review established that gut dysbiosis produces neurological, immune, and metabolic effects far beyond the GI tract. The gut microbiome diversity research of the past decade has converged on a single most powerful predictor of microbial health: the diversity and quantity of dietary fiber consumed. Sonnenberg et al.'s research showed that different bacterial species specialize in fermenting specific types of fiber, meaning variety, not quantity alone, is the critical variable for restoring and maintaining microbiome diversity. The American diet's fiber deficit is one of the most well-documented nutritional problems in the country. The average American consumes approximately 15 grams of fiber per day, against a recommended 25 to 38 grams. More importantly, fiber type diversity is even more depleted than total quantity, driving the microbiome impoverishment Sonnenberg described. Different dietary and lifestyle interventions produce distinct levels of microbiome improvement, as measured by diversity indices and beneficial bacteria abundance. The research allows a clear ranking of the most effective gut repair interventions. Gut repair is not a single intervention but a multi-step process that addresses the intestinal barrier, microbial diversity, and the systemic effects of dysbiosis. Understanding each step guides the sequencing of the repair protocol. The evidence-based gut repair framework follows a 4-phase protocol: remove, restore, re-inoculate, and maintain, adapted from functional medicine's Fora model and grounded in current microbiome research. Gut repair follows a timeline that depends on the degree of dysbiosis and how consistently the protocol is applied. Microbiome composition can change significantly within days of dietary change, but full restoration of diversity requires sustained effort. The daily implementation of the gut repair protocol requires 6 consistent actions spanning dietary diversity, fermented foods, targeted supplementation, and behavioral factors that affect the gut-brain axis. Gut repair protocol must be individualized based on the nature and severity of dysbiosis, prior antibiotic use, and specific symptoms. These adjustments match the protocol to the individual's gut health status. The science on gut microbiome health is clear, and the most effective interventions are dietary, not supplement-based. Here is what the research shows. The evidence is clear. The mechanism is understood. And the steps to rebuild your gut are within reach, for anyone who addresses the actual drivers. Follow MedBrief today for more science-backed health insights, explained clearly.